only interact with a small subset of the available motifs that are encoded in a genome, often as little as 1 to 5%. And what's more is that as you compare the same transcription factor in different cell types, they can often interact with different subsets of those motifs. And so despite sort of the certainty that we get from these biochemical mechanisms, if we want to ask, what can a transcription factor do, we have to invoke some sort of a vague concept of biological context to explain this. So there's a lot of potential determinants of this biological context, but I'm particularly interested in one of these, which is that it has to do, in some ways, with the way that DNA is packaged in the nucleus. And so, as we all know, DNA isn't just packaged as a naked molecule in the cell, but it's as part of a large ribonucleoprotein complex termed chromatin, whose primary function is to impart a high degree of compaction to the DNA molecule so it can fit into the relatively small eukaryotic nucleus. And in order to compact the genome, this comes at a cost, which is that encasing DNA in these nucleosome core particles, and even in this least compacted state, has the effect of effectively masking the available information encoded in the genome. And so on a very small scale, the presence of a nucleosome over some of these motifs can effectively mask that information from being read out by these regulatory factors. And so you can see how on a very small scale, just the organization of chromatin structure in a cell can potentially have a large impact on the way that these regulatory networks function. But we also know that chromatin structure is itself fluid, right? That there exist mechanisms for driving changes in nucleosome positioning. And so we can imagine now perhaps changing the location of these nucleosomes at tens or hundreds or thousands of sites across the genome can in principle give rise to a diversity of responses from single input generic transcription factors. And so in my lab, we're interested in studying really the interface between these two systems to ask how regulatory networks are constrained by their underlying chromatin structure, but also to ask how biological systems deploy changes in chromatin accessibility in order to give rise to these diverse responses. So the way that I like to study this is by understanding the development of the fruit fly Drosophila melanogaster. And so for those of you who are less familiar with this system, these images show stills taken from a movie every 3 hours over the 24-hour period of fly development. And the embryo starts off as a relatively simple system where this football-shaped thing here is the embryo, and all of the cells at this 3-hour time point are located on the outer cortex of the embryo in a simple epithelial sheet. And these cells at this time are essentially unrestricted in their developmental potential. And as they develop, each of these cells will be subjected to a stereotypical sequence of events in the form of biological signals, patterning events, that will convert each of these unrestricted cells into one of the very specific cells that comprises the larva that's formed at the end of this process. And so studying this question of the interface between regulatory networks and chromatin structure, this system gives us numerous advantages to studying that question. And one of those is that we can, that development gives us a natural sort of time axis, right, where events occur in a stereotypical order. Another advantage is that we have a vantage point on the very starting state of the system. And so, for instance, at this moment in development, before these differentiation events have occurred, we can look in each nucleus and we can see that approximately 10,000 regions define the total of open and accessible chromatin in each of these nuclei. So these comprise the sum total of all of the available enhancers, promoters, and insulators that are encoded in the genome, or accessible information. What's more is that decades of research on the fly development have identified all of the factors that are necessary and sufficient for driving the very first decisions that will convert these cells into their numerous ultimate cell fates. And so these here are factors that are either transcription factors or signaling pathway components. And the way that I think about it is that these initial 10,000 regions represent the limited regions in the genome where these factors can initially interact in order to begin eliciting these distinct fates. And so in today's talk, I want to tell you about two stories where we've been harnessing these sorts of advantages to understand two questions. One is how is that initial sort of ground state of chromatin structure built? And how far in development can you get given that initial ground state? And how do changes ever arise as cell type specific changes are installed? And so, before embryos have the ability to really fully engage in this process of differentiation and morphogenesis and commitment to particular cell fates, they first have to undergo a series of 13 rapid synchronous mitotic divisions that serve to amplify the single nucleus that's formed after fertilization into the approximately 6,000 nuclei, part of which you can see here, all nicely arranged on the outer cortex of the embryo. And it's only after completing these divisions that embryos then begin the process of morphogenesis, differentiation, and patterning in earnest. And so this transition from the initial proliferative state to one of differentiation comes at a major developmental milestone termed the mid-blastula transition, or MBT, which is happening right now. And at the MBT, we have several critical biological regulatory systems that are undergoing a tremendous change in their function. So for instance, here in this movie of histone GFP, you can see very clearly that embryos have, or that the nuclei have all sort of paused their cell cycle. The early cell cycle is extremely fast, consisting of very brief 10-minute to 20-minute cycles. And at the MBT, the cell cycle gains a gap phase and now can take up to at least an hour before the next mitosis. What you can't see in this movie is something that I think is more interesting, which is that over the course of this movie, each of these nuclei is becoming transcriptionally active for the very first time in a process called zygotic genome activation. And so at the MBT, thousands of genes will bind RNA polymerase for the very first time and begin transcribing the very first transcripts produced from the embryo. Now, this process isn't just a matter of switching genes on, but instead represents a more fundamental change in the underlying chromatin structure that supports transcription. And so if I were to shrink you down and to drop you inside one of these nuclei very early in development, you would be hard-pressed to find some of the features that you would expect to see in the average eukaryotic nucleus, including widespread open chromatin and promoters. You wouldn't notice any, there's limited histone modifications. The differences between euchromatic and heterochromatic compartments have yet to be defined. And there's not much in the way of higher order looping and structure of the chromatin at this time. All of these features are acquired more or less at this time in development. And so we can use this system basically to ask, how do these fundamental features of the genome get built from the ground up? And so when I was a postdoc, I became interested in the following problem, which is that in order to undergo zygotic genome activation, these nuclei need to build these features of chromatin structure. But they do so in a time of extreme mitotic activity. And neither of these two periods of the cell cycle are particularly amenable to establishment of these stable and heritable features of chromatin structure that the embryo needs to build. And so what I mean by that is that in order to achieve such a rapid cell cycle, the early embryo uses a cell cycle that is essentially stripped down and built for speed. It consists entirely of M phase followed by S phase without any intervening gap phases. And neither of these periods of the cell cycle are intuitively amenable to establishment of these features of chromatin structure. So for instance, we have this tremendous change in chromatin conformation that accompanies mitosis, and often we see eviction of regulatory factors that control transcription and chromatin accessibility. And similarly, during DNA replication, we see that, or we know that DNA polymerase coming through chromatin will displace nucleosomes and essentially scramble their position. And so at the very least, this rapid cell cycle serves as an impediment to the faithful inheritance of this information over cell cycles or over short periods of time. However, if we quantify cell cycle dynamics in the embryo by measuring either mitoses or quantifying DNA replication by use of this reporter, we get a picture that looks like this. And so I'm showing you the last 3-cell division before the MBT is complete at this time. Mitoses are in gray bars, and our estimate of DNA replication activity is shown in blue. And what you can see is that over these last 3 cell divisions, the cell cycle is undergoing remodeling, right? The cell cycle is roughly twice as long at cycle 13 than at cycle 11. And if we look at DNA replication, whereas the entire interphase period in cycle 11 is consumed with replication, at cycle 13, now we have perhaps 2 populations of early and late replicating domains beginning to emerge, as well as a brief, perhaps post-replicative G2-like stage beginning to happen. And so what we'd like to ask is when is there time in this cell cycle to build open chromatin states? And ideally, we'd have a tool like this that we could quantify and just overlay on this plot. But there is no really good way to do this on a genome-wide basis. And so as a postdoc, I did really the next best thing that I could think of, which was to use a then new technique called ATAC-seq in order to make measurements of nucleosome positioning and accessible chromatin states in single embryos that I could collect at 3-minute intervals over the 3 cell cycles leading up to the MBT. And the design of this experiment was such that we would take, that we expected that over this 42-minute period, we would see acquisition of that initial ground state of chromatin structure by the end of this time point, and that with this temporal resolution, we should be able to ask how these regions gain accessibility and are maintained with respect to ongoing cell cycle progression. And so when we do this experiment, we get genome-wide measurements at all these times that look like this. And so I'm showing you an example here over a gene called hunchback, where I'm showing you about 10 kb of genome over here surrounding this gene. And the degree of chromatin accessibility is indicated by the height of these peaks that are in navy blue at each of the time points listed on the side. And so hunchback is a gene that is one of the rare genes that is actively transcribed during this period. And you can see that essentially, from as early as I've looked here at nuclear cycle 11, we can detect accessible chromatin at critical genomic features like this P2 promoter, as well as this associated shadow enhancer. Regions like this, though, turn out to be a minority with respect to the entire sort of profile of regions that are open by the end of this time point. In contrast, so only about 30% of these regions, of the regions in the entire ground state, gain accessibility this early. The remaining 70% of these regions dynamically gain accessibility as embryos approach the MBT. So for instance, this gene Skilla has a promoter that's initially inaccessible, and that by prophase of nuclear cycle 12, like clockwork, something has happened that confers chromatin accessibility at this promoter for the very first time. And so what this data allows us to do is to get a vantage point on the relative timing of the acquisition of these chromatin states, and I'll have something to say about the relative timing between these 2 groups in a couple of slides. But before I get to that, I also want to point out one notable feature that really jumped out at us is that we intentionally collected 3 time points where each nucleus is in metaphase. And what we found is that despite this tremendous conformational change that accompanies entry into mitosis, that these regions nevertheless maintain full chromatin accessibility at the level of these individual regions in the genome, essentially at every locus that we looked at. And so this tells us that despite this huge change in chromatin conformation, at the level of individual nucleosomes, some mechanism exists for maintaining the accessibility of these regions, and perhaps indicates the existence of some form of bookmarking for maintaining these regions. It also tells us that with respect to establishment and maintenance of these states, that mitosis itself isn't an impediment to the faithful inheritance of this information from one cell division to the next. If you look carefully, you can see a minimum at each cell cycle corresponding to the earliest stages after mitosis, and this corresponds to an initial burst of DNA replication that occurs in these embryos. And so in general, we see that there are minima during DNA replication that are eventually overcome by the later half of each cell cycle. And so in general, we're interested in understanding both what the factors are that drive the initial acquisition of chromatin accessibility at these regions, as well as the mechanisms that maintain accessibility over mitosis. And so to address the first question, One thing that's very straightforward to do is to just take this entire set of data for all 10,000 regions and ask what are the most enriched motifs when we, within these open tracts of DNA. And when we do that, we get a result that people in this field have been getting for about 20 years, right? Which is that the two most enriched motifs in this data set correspond to binding sites for two now well understood transcription factors. Here we have CAG-TAG, which is the binding site for a pioneer factor called ZELDA. And here's this non-complex GA repeat sequence that is the binding site for at least a trio of transcription factors, GAGA, CLAMP, as well as PIP-SQUEAK, and perhaps others. And so I said that the same result's been coming up for years and years is because these are the two most enriched motifs that you get if you, for instance, ask what are the earliest expressed genes in the embryo, or where are my favorite enhancers. These are present everywhere. The idea, so I introduced these as pioneer factors, and because many of you are in Melissa's lab, you know what I'm about to say, so, but I'll pretend like you're not here. So as pioneer factors, we imagine that these are factors that have a special ability to bind to these motifs, even if they're encased in nucleosomes. And by doing so, they can serve as essentially the first factors to bind, and somehow, by mechanisms that we don't fully understand in many cases, establish greater regions of open and accessible chromatin in the neighborhood of these binding events. And so we can visualize this in our data. So if we just take each of these motifs and plot the average nucleosomes that we can calculate from the ATAC-seq data around each of these motifs, what we see is that on average we get about 170 to 260 base pairs of general open and accessible chromatin around each of these 7 base pair motifs. And so this here is just a plot of the average nucleosome distribution, where you can see clearly this anti-nucleosomal signature centered over the motif. In the case of ZELDA, we were able to test directly, as many others have done, including Melissa's lab, as in Julia Zeitlinger's lab, and Chris Rushlow, and confirmed that ZELDA is in fact required for displacing these nucleosomes away from these sites. So if we just repeat these experiments in a ZELDA mutant embryo, we see that now we completely lose this open chromatin signature, where the nucleosomes essentially collapse back upon themselves over these motifs. And so this confirms that ZELDA, presumably by binding to these motifs, can establish general regions of accessibility around these sites on a very small scale. But collectively, if we look at the entire sort of profile of accessible chromatin in the early embryo, where we take all 10,000 of these initial regions and stack them up on top of one another in this heat map, we can see that compared to wild type, where all these regions are open, that in a ZELDA mutant, we lose accessibility at approximately 30% of these sites. So this tells us that pioneers like ZELDA are playing an outsized role in establishing at least components or subsets of accessible regions within the genome. Now, recently, Melissa's lab, as well as Erica Larchan's lab, have looked into the role of GAGA and CLAMP onto contributing to the remainder of these regions here. And I think surprisingly, is that they actually, it's not like GAGA and CLAMP account for the rest of this. And so overall, I think that that there still remain other factors that are operating in the early embryo that are contributing and working together to establish these open chromatin states. And so with respect to timing, I'll just quickly go through this because it's published, of course. With respect to timing in these two classes that we saw, where some regions were opening up early and some were opening up later, by at least motif enrichment, we found that there was a very strong enrichment for early regions to be bound by ZELDA, and these later regions to be bound by GAGA as well as CLAMP. And so this suggests that in terms of the timing of how this initial ground state is being built over time, that this relies on to a certain extent on the timed deployment of these individual pioneer factors to sort of build the system in a stepwise manner. We plot on average the accessible chromatin profile across these time points, now in reference to our DNA replication estimate. what we see is an indicator that basically as the embryos pass through these cell cycles and are subject to the influence of these pioneers, that they make gains in chromatin accessibility that are biased towards the ends of each of these cell cycles following the passage of the replication fork. And that some mechanism exists to maintain that information more or less from one cell division to the next. And so overall, these are our rules that we're operating with moving forward to understand the system better. Namely that we want to identify pioneer factors that at least do some of the work, and to understand the timing mechanisms that control their timing of activity, and to focus on how this work is remembered over mitosis. And in particular, for the second point, we're particularly interested if the sequence of events is important for anything at all, right? This could just be a secondary consequence or it might be irrelevant, let's say, to the normal functioning of an embryo. So we've done a little bit of work, or we've started a little bit of work over the pandemic on understanding how this work is remembered over mitosis, and so I wanted to share this. And so by reference to literature as well as through a genetic approach that I don't have time to explain, we had a suspicion that the compass complex, specifically the form of the compass complex that contains set1, could be functioning as a candidate bookmarking factor. This is based on the fact that Gerd Blobel's group had shown a few years back that mammalian versions of the compass complex do have bookmarking ability, and I believe an immune cell type. And so we became interested in testing directly if this complex could also be playing this role in early embryos. So what is this complex? It's generally required for the transition from initiation to elongation in in general during transcription, as well as it's responsible, particularly the set1 compass complex, is responsible for the vast majority of histone H3 lysine 4 di and trimethylation. And so this is a modification that tends to correlate with active gene expression and typically marks 5' ends of the genes where this initiation to elongation transition is occurring for POL2. In addition, back in 2013, Julia Zeitlinger's group had shown a really dramatic switch in the K4 methylation state of the set of promoters that become transcriptionally active during zygotic genome activation, where these are heat maps over a set of the first zygotic genes that are transcribed, looking at POL2, TBP, as well as K4 trimethylation. There's a small set of POL2 genes, or POL2 bound genes that are transcribed before the MBT that have TBP, but remarkably don't have lysine 4 trimethyl marks. And that following the MBT, now we recruit POL2 to thousands of new genes. Many of these are marked with this K4 trimethyl mark, at least if they're actively transcribed and not poised. But notably, this set of very highly transcribed early genes doesn't really accumulate K4 methylation substantially. And this has always kind of been just a weird feature, both the timing, this beautiful switch-like change in methylation status, as well as the special status of these genes. So we decided to have a look and see what's going on with set1. And so we've managed to introduce a GFP tag to set1 and to use that to study its distribution, both genomically as well as by microscopy. And so if we do ChIP-seq for set1, what we find is that it is basically present at any gene that is transcribed, which is a good thing, perhaps not surprising. So for instance, here's this E-cadherin shotgun that has a nice RNA polymerase peak at the 5' end, some polymerase within the gene body. Set1 nicely localizes just downstream of this pause polymerase peak, as well as distribution of the di and trimethyl lysine 4 mark, as we would expect to see. Kind of like a textbook view of what these modifications should look like. So this is encouraging, but interestingly, what we found is we were able to confirm Julia Zeitlinger's result that if we look at one of these really highly transcribed early genes, this is a gene called SLAM, that we don't see any of this K4 modification here. But weirdly, we see reproducibly and specifically the binding of set1 to this gene. And so we think that what this suggests is that this enzyme isn't strictly limited to the sites where these modifications are taking place, but maybe this is an indicator that SET1 is playing a broader role in transcriptional control even of these really highly expressed genes, which would be great. Now remember, if these genes are amongst the earliest expressed genes in the embryo, and so we got very excited to look at what SET1 looks like in an embryo in terms of its localization. And that's where we got kind of a surprise. And so this is a movie of SET1, GFP, and that's just in this channel here, and then this is that channel merged with histone RFP. The movie starts off in metaphase of nuclear cycle 10, as you can see here, and I just say ignore these really bright spots, which is yolk autofluorescence. They go away really quick. So I'll just let it play, and you should be able to see what's weird about SET1. Is that initially, it's completely excluded from the nucleus. At cycle 12, we begin to get little foci. Cycle 13, we get more foci. And if you can tell, the relative concentration of SET1 in the cytoplasm is getting drained out and pass it into the nucleus. So at this point, you can positively detect where the nuclei are simply by the SET1 fluorescence intensity. And so this argues against a general role for SET1, in regulating broadly the expression of these early genes, since many genes are expressed before SET1 is even robustly located to the nucleus. But it highlights the fact that perhaps the timing of that K4 methylation event is strongly linked to when SET1 is available to actually impart that modification in the nucleus. And so this is just a quantification of the nuclear concentration of SET1 over time. And I'm floored by the fact that you could probably draw a straight line right through the interphase data points here that suggests that this enzyme is subject to a really slow process of nuclear import into the nuclei. And it highlights for us what I think is a frontier in terms of thinking of the timing of these early embryos, and reluctantly pulls us into the question of thinking about nucleocytoplasmic shuttling. And you never know what you're going to end up studying when you look at these things. But we think it's important because if we look at other pioneers such as GAGA factor, which has recently been shown by the LAGA lab to also have a bookmarking-like function for at least some of its functions, at some of its targets, we see that nevertheless GAGA also shows a distinct sort of graded import into the nucleus. If we were to measure these concentrations over time, it would not look unlike the SET1 data that I just showed you. And so there is some overall sort of control of regulated nuclear localization that is playing a major role with respect to the apparent timing that we see for these changes in chromatin accessibility. And so, let's see. So overall, right, we've focused initially on understanding the establishment of this ground state, partly because it's an interesting developmental biology problem. How do you build the first state of chromatin structure? It appears that we're now thinking about biological timers that control the nuclear localization of these factors, trying to identify additional chromatin factors that are, or pioneer factors that are imparting these open states, and searching for functional evidence on how these factors are, how these regions are remembered over mitotic divisions. And so that's the developmental biology side of things. But we can also, well, it's all developmental biology, but the more sort of gene regulation aspect of it has to do with, I think, the picture that this paints of the starting state of the embryo and what we can do with that. So what I mean by that is that everything that we know that regulates the initial sort of ground state of chromatin structure in these embryos is a factor that is expressed everywhere in the embryo. So Zelda is present equally as much at this end of the embryo as it is over here, as are all of these other factors. And so we can imagine that that initial state of chromatin structure is homogeneous or shared by all of the nuclei in this embryo. And so we can now turn to the question of how do any changes ever arise, and what do embryos do with this information in order to begin giving rise to cell type specific patterns of gene expression. And so we can imagine, for instance, that as the spatially restricted cell fate determinants become activated in one portion of the embryo or another, that again, these initial regions that are established at the beginning of this process serve as the limited set of regions that can be interacted with by these factors that will then direct cell fate decisions. But once we had this data, we sort of realized that we could do this a bit more systematically. And instead of sort of pointing at this in the abstract, to actually dive in and look at this mechanistically based on the way that we've, based on nearly 40 years of research on the development of this system. And the reason for that is that, as I said before, a lot of effort has gone into identifying genetically the factors that are required for these very early developmental events that, for instance, segment the embryo into the 14 segments that define the future head, thorax, and abdomen of the fly. And so, for instance, here, the way that that's thought to work, and that appears in every developmental biology textbook, is that embryos start off with graded information provided by mom. So for instance, here, this transcription factor bicoid is presented as a gradient that's strong in the anterior and weak in the posterior. And this has the ability to impart positional information along the long axis of the embryo. But remember, the embryo can't respond to this information until zygotic genome activation takes place. Once it does, bicoid activates a set of 3 hierarchically related gene regulatory networks that convert this graded information into the 14 segments that define the different regions of the larval body plan. So first off, we have gap genes that take this gradient and turn on in distinct blocks of expression across the axis. The gap genes then work together to activate expression of pair rule genes, which are activated in distinct 7-stripe patterns, each demarcating pairs of segments. And then finally, the pair rule genes become activated, function to establish the expression of segment polarity genes, which become active in individual rows of each segment, delineating, say, the posterior or anterior half of each segment. And so not only do we know all the players in this system, but we also know all of very many of the cis regulatory elements that are responsible for making these wonderful gene expression patterns. And so we did kind of just a thought experiment. We said, well, we start off with this initial ground state. How far can you get in development with just that information in terms of accessible chromatin in terms of this process? And I should say that whereas these zygotic gene regulatory networks begin their function around zygotic genome activation, this process is largely in place in terms of the initial expression of these genes by about 1 hour later in development as embryos start gastrulation. So can we look and see how far they can get in this process? And what we find is that in terms of accessible chromatin that's available, the embryo can get about halfway through that process, right? And so the way I'm showing you that here is I'm showing you the chromatin accessibility at shortly after zygotic genome activation for one member of the gap genes, one member of the pair rule genes, and one member of the segment polarity genes. And I've highlighted in red the enhancers that are open at this early time. So for instance here, giant, which comes on in these unique patterns, the known regulatory elements that control giant expression are both open and accounted for at ZGA, as we would expect. Pair rule genes, which come on in 7 stripes, have this unique regulatory sort of configuration where they initially turn on due to the activity of individual enhancers that control single or pairs of stripes, and then later on hand off control to later acting enhancers that control all 7 stripes together, like zebra elements is what they're referred to. And when we look at the accessibility state of those loci at ZGA, we see that all of the early enhancers have gained accessibility nicely, but the late enhancer has yet to gain full chromatin accessibility. It gets even worse when we go to the segment polarity network, where many of the known enhancers for engrailed have yet to gain full chromatin accessibility. Now, if we look 1 hour later, once all of these networks have become active, and some have sort of even moved past their initial activity, what we see is that additional changes have taken place. So now measuring it at gastrulation, what we see is that now we've lost some accessibility at some of the gap gene enhancers. We've now gained accessibility at the Eve late enhancer, and we've gained accessibility within these regulatory elements at engrailed. And so this tells us that even after ZGA, there continue to be changes in chromatin accessibility, which in itself isn't surprising. As I said, chromatin structure is itself fluid and will be constantly subject to change. But when we look then and say, well, do the rules that apply to that initial ground state also apply here? Namely, do we see Zelda binding to these regions here, or Gaga or clamp? And the answer is not really. So when we look at the sort of the usual suspects for regulating the ZGA state, we see that very few of these enhancers, for instance, at the engrailed locus, are occupied robustly by these ZGA era sort of pioneers. And so this tells us that in order to drive these changes that are occurring later in development, just a bit later in development, this likely requires additional, as yet unidentified, pioneer factors. And we set out to try to identify some of them. And the approach that we took was basically to ask, well, here we have a 1 hour period in wild type embryos. Let's look and see and characterize all these regions that gain accessibility or lose accessibility over time. But that's not enough. We want to ask, basically, is this change occurring just as a matter of the passage of time? Or is it occurring in a likely spatially restricted manner by virtue of receiving input information from the embryonic patterning systems that dictate these different sort of compartments of the embryo? And so we decided to compare wild-type embryos with embryos that completely lack all maternal inputs to embryonic patterning. So this is an experiment that we can do uniquely in the Drosophila system, where, again, through genetics, we know all of the maternal factors that basically provide the positional information to distinguish each cell type along the primary cardinal axes of the embryo, giving rise to distinct anterior, medial, posterior gene expression states, as well as along the dorsal-ventral axis. It's possible to make a complex mutant that knocks out or flattens the gradients that provide this information. So for instance, here this is a bicoid Oscar Capicua torso-like toll RM9 mutant. They're all on one chromosome, so it's just one cross after a lot of recombination. And the remarkable effect of this is that we generate what I'll refer to as patternless embryos, but they're not exactly patternless. What it is that we've converted every cell in the embryo to have the gene expression profile of just a small subset of cells in a wild-type embryo. It's like a very low-resolution single-cell experiment, where here now this genotype yields embryos that are entirely derived from posterior and lateral fates. And by reference to a real single-cell RNA-seq experiment, cells that express both of these markers correspond to a small population of about 60 cells that will ultimately contribute to the posterior endoderm, or the posterior gut of the embryo. So now we can ask, when we see changes happening in the embryo, are they also happening in this genotype, or are they affected in some way? Where we'll expect losses or changes in the negative direction if there's a localized change occurring outside of this region, and an enhancement in the effect if we measure change that is restricted to posterior endoderm. So there's a number of ways to summarize these data. I've chosen PCA for you today. And so this is the PCA of this experiment, where each dot represents a replicate measured by ATAC-seq. The labels here are good. And so what we find is that the primary source of variance that is driving changes in chromatin accessibility between ZGA and gastrulation is occurring independently of patterning, namely that it's time dependent, let's say. As these embryos mature, additional regions are gaining or losing accessibility independently of any spatially restricted information. However, there is a significant contribution secondarily from positional information, where we see that the contribution from patterning systems is less so at zygotic genome activation and is much more pronounced during gastrulation. And so this tells us that there's inputs of both types into these sort of timed changes in chromatin accessibility. And what this allowed us to do was to categorize 10 different types or classes of chromatin element that either gain accessibility or lose accessibility dependently or independently of patterning. So for the rest of this talk, I want to focus on just one of these classes, which I'll introduce in comparison to another one. So for instance here, if I plot the scaled change in accessibility over time by genotype, here's one class where they gain accessibility in wild type but not in the mutant, and sort of then its conceptual opposite, where it gains accessibility in the mutant more so than it does in wild type. Here's an example of what this data looks like. And so if we look here at the sloppy paired cis-regulatory module shown here in red, it gains this lovely sort of broad peak of accessibility by gastrulation in a wild type embryo, but fails to do so in a mutant. And conversely, within this toll-like receptor, we find that this region here dramatically gains accessibility in the posterior, in the mutant at gastrulation. And we can clone each of these enhancers, put them into a fly, and we find that this is expressed within posterior endoderm, and this is expressed outside of posterior endoderm, as we would expect. So again, doing our trick where we look for enriched motifs, what we find is something really encouraging down here, which is the enriched motifs in this case correspond to two orthologs of characterized mammalian pioneer factors. FOXA, which I guess could be the proto-pioneer factor, the first one, whose ortholog is forkhead, as well as brachyury, which around the time that we published this had been identified as playing a pioneering role during mammalian mesendoderm specification, who's known as brachyentron in Drosophila. These factors are expressed in the posterior endodermal fates, and we think that this is consistent at least. It suggests that perhaps they're functioning in a pioneer-like manner to confer these changes in accessibility, specifically in this small subset of cells. What's striking to us is that when we look at this class, the enriched motif corresponded to this factor called odd-paired, which is the ortholog of the mammalian ZIC1 through 3 proteins. And as far as we know, this is both a really understudied protein in Drosophila, and let's say lacks deep mechanistic understanding in the mammalian literature with respect to its function either in development or in other contexts. But notably, odd-paired is a member of the pair rule gene regulatory network itself, and it has this really weird expression pattern of a sheath around sort of the middle segmental primordium of the embryo. So like I said, odd-paired has received relatively little attention in the history of fly developmental biology, and sort of similarly within developmental biology in general. And one of the reasons is that although it has this unique role in being necessary for specifying pairs of segments across the segmental primordium, unlike the other 7 members of the pair rule network, which are activated in a beautiful 7-stripe pattern, it just comes on in this sort of boring sheath pattern. And so these factors have naturally received a lot more attention, primarily because it's a conceptual challenge to turn on gene expression in stripes. Another reason that distinguishes odd-paired from the rest of the members of its network are that it is expressed late. So for instance, this is a timeline of nuclear cycle 14 where ZGA is occurring off the scale over here. And whereas runt and eve are expressed nicely during this early period and throughout this period here, odd-paired shows a distinct delay in its activation of about 30 minutes before we can detect both protein as well as transcripts. And so it comes on late, it's harder to study. However, Eric Clark, when he was a grad student with Michael Acom at Cambridge University, took a really careful look at the way that odd-paired functions in terms of genetics, and found that it's required for some, if not all, of the late pair rule enhancers to become functional in the embryo. We can visualize that if we just make a reporter of the Eve late enhancer that I showed you before, and just plot its activity in real time relative to the expression of odd-paired. And you can see with the exception of the very first stripe, the rest of these regions become transcriptionally active around the time that we detect odd-paired expression switching on in the middle of the embryo. So odd-paired is required for the function of this enhancer, but it's not doing this as an activator per se. We think that it has a more general role for driving changes in chromatin accessibility, which we can see here. So if we do chromatin IP for odd-paired, we see that it nicely binds to the Eve late enhancer. But now when we do a TAC-seq comparing wild type and odd-paired mutants, we see that odd-paired is completely required for driving full chromatin accessibility at this site. We can generalize this if we take all of the regions that are bound by odd paired and look at the effect of mutating odd paired via attack. We see that similar to what I showed you for Zelda, that about 30% of its targets require odd paired for full chromatin accessibility. And if we look at the molecular sort of addresses of these factors, we find that they're strongly overrepresented for components of the very gene regulatory network that odd paired participates in, including many of the late-acting enhancers within the pair rule genes, as well as many of the late-acting enhancers in the segment polarity network. And so we think that this data basically gives us an interesting handle on thinking about gene regulatory network function within embryonic patterning, but just in general, which is that we tend to think of these systems as being driven by either positive or negative transcriptional regulation of target genes. We tend to draw these things like this. However, we know in the case of this pair rule network that it undergoes spatial changes in its expression over time, where, for instance, paired initially is activated in a 7-stripe pattern, which eventually matures to this 14-stripe pattern by repression of a row of cells in the middle of each stripe. We have no way to account for this if we don't incorporate both a time axis to this regulatory network, as well as now knowing that odd paired is functioning as a pioneer, we have a potential mechanistic way to approach this, suggesting that the critical repressor for generating this 14-stripe pattern, although it may be expressed, may not be able to interact and regulate this gene until odd paired comes along, opening up this enhancer and now allowing for this repressed center stripe pattern to occur. And so we're currently undergoing or testing this for the entire network, basically hypothesizing that there must exist over time, over this 1-hour period, restricted or DNA binding events for these factors that are dependent on the underlying chromatin accessibility state. And so we hope to trace all of the odd paired dependent binding events within this network in order to formalize this, as well as test the rest of the factors, the rest of the 7 factors in this network for whether they influence chromatin accessibility positively or negatively. But most generally, what we think is that if we can get away from this idea of simply thinking of transcription as being a positive or negative regulation at the level of transcription, and consider that transcription factors can also influence how information is read in the genome, this can solve a problem of being able to get maximal information from a limited set of input components to a network, where in the case of odd paired, by driving changes in chromatin structure, this allows for elaborations on an initial spatial pattern. So in the last couple minutes, I want to return to one question. So as I've told you, I've been sort of focusing on this changes over time, namely how in the first part of the talk we described a likely early wave of Zelda-dependent chromatin accessibility, later waves of GAGA and clamp-dependent chromatin accessibility, and now I've told you about a later-acting zygotic transcription factor that is working to elaborate on this initial, mostly homogeneous chromatin state. This is a sequence of events, and again, this, we kind of wonder, like, okay, so all you're measuring is whether there's open chromatin, but is it playing a deterministic role? Is a question that's been really hard to address. Does this matter at all? Or if we, for instance, were to provide all of these states from the very beginning, would that have any impact at all? And so this is really hard to test with these 2 factors for technical reasons. But our identification of odd pair now gives us the unique ability to test this question of timing. Specifically because, remember, odd paired is expressed late, and it's zygotic. And what we figure is that, well, if we were to look at chromatin accessibility before odd paired is even expressed during nuclear cycle 13, if you think about it, it should look just like the chromatin state in an odd paired mutant. And that's what we see. So if we now measure approximately 80 minutes before this time point, this is nearly indistinguishable from the odd paired mutant state. And so then just using a fairly straightforward transgenic approach, we can now introduce odd paired maternally at the same levels that it's expressed in a wild type embryo zygotically, but just at an earlier time. So a true misexpression. If we now provide odd paired at this time, what we see is that dramatically it's able to confer open chromatin accessibility at these targets. At almost all of them, not entirely all of them. So again, we see this effect that a region like this enhancer downstream of the odd skipped locus is bound by odd paired. It requires odd paired for chromatin accessibility at gastrulation, and that we can drive its accessibility prematurely by maternal expression. Naturally, we want to ask, well, what does this do to embryonic development? These embryos, depending on how much you drive odd paired expression, will have subtle defects in their embryonic patterning, but reproducible ones. And so what we find is that if we make a reporter out of this region that's highlighted here, in a wild-type embryo, it turns on in this beautiful multi-stripe pattern across the embryo with a regular stereotypical spacing, indicating positions along the segmental primordium. However, if we prematurely express odd paired, we get this very satisfying result that the reporter turns on earlier, not shown here, and when it does, it turns on in the wrong pattern, namely that now this gene here, where it's controlled by this enhancer, is now being turned on in cells where it should never be turned on, and too early. So this is, to me, the best evidence that it's not just a matter of measuring changes in chromatin accessibility for their own sake, but that the sequence with which these events occur is actually critical for the overall precision of assembling a body plan for an organism. And so with that, I've mentioned some things that we're continuing to work on, but overall I hope that I've given you a good introduction to our thinking on how this initial ground state is being built and how it really sets up the embryo for undergoing the very first stages of embryonic patterning, and how we've been using this system to begin investigating mechanisms for giving rise to these cell fate-specific chromatin states, and the significance of doing this in the right time in the right place. So with that, I'd just like to acknowledge my lab, and particularly Natalie Gilbert, who did the set 1 work that I was showing you earlier. Collaborators, willing or otherwise, listed here, as well as my funding. And I'd be happy to take any questions.
Okay, go for it, and then I'll...
So in the case of the factor, and I can't remember which one it was that was showing slow nuclear accumulation, there's a nuclear membrane breakdown in each mitosis, correct?
There is.
So what I'm wondering is if you're more looking at chromatin association than actual nuclear import.
Yeah, so let's see. So with respect to, there's nuclear envelope, there's an incomplete nuclear envelope breakdown in Drosophila mitosis, and we do see the factors flood back in to occupy, sort of co-occupy, at least in confocal space, with the chromatin. We don't know if that's a productive binding or just sort of local occupancy, and so we can test directly the binding by specifically by harvesting embryos that are in mitosis. And so we might expect to see accumulation. So for GAGA factor, a different lab, Munialaga's lab, has demonstrated that if you fax purify nuclei that are in mitosis, that you can detect GAGA bound to chromatin at that time. And so I'm not sure if that gets to the heart of your question, but it is a little tricky to just judge from the microscopy. Yeah. In the odd paired movie that I showed really quickly, we can see very clearly that odd paired protein is nicely associated with mitosis, mitotic figures. And so this is thought to be a feature of at least some of the pioneers that are out there, but it isn't a required function. So for instance, Zelda doesn't do that, as you've probably heard from Melissa.
I'm going to ask you a question about the set 1. So you made the argument about the nuclear import, but I think that the SLAM ChIP-seq, right, and then Julia's data show that H3K4, even though set 1 is there and nuclear cycle 14, it's not methylating, right? Yeah. So what's your theory there?
So so if you look at that class of genes, it's almost all of them are really short-lived in terms of time. So they're expressed at, they have as much pull-to on them as they possibly can, and then at some point during nuclear cycle 14, they get shut down. they get shut down both at the level of losing polymerase, but in the data that I didn't have time to show, they are universally losing accessibility at the level of attack. So we've become kind of interested in those because nobody ever studies why genes turn off, right? We're only focusing on why they turn on. And those are remarkable because they have this really tight temporal role. So we imagine that one possibility is that because SET1 is there, the K4 methylation could be established, but perhaps it's being removed at the same time because of some regulatory mechanism. I'm not a fan of that type of futile stuff. However, it's also possible that something about the regulation of the gene is preventing the K4 methylation from being established, and that this somehow allows these genes to be more susceptible to shut down more rapidly. So, we kind of hypothesize that there comes a time in the life of every embryo where they begin actually responding to the information that these histone modifications, are promised to provide to transcription. And it's possible that sometime in the middle of cycle 14 is when those repressive mechanisms kick in.
I remember way back with the Lee lab with the Lee et al. Paper, talking about the H3K4 trimethylation, therefore not being required for transcription.
Yeah.
Yeah, well, I was also wondering if the catalytic activity of SET1 is important for any of that, but also, SET1 is great because it has this crosstalk between chromatin modifications, and you have H2A UB, right, that is helping direct it, and what's directing that is K79 methylation. So I'm wondering what's known about those modifications prior, and could that explain some of the, oddities in your data?
Yes, we haven't looked at the enzymes for the ubiquitination of H2B, but I asked Natalie to give me the simplest slide, and so we've also looked at the ubiquitination mark at H2B, and it correlates perfectly with the K4 methylation and not with the SET.
So those 30-ish genes at the top that never acquire K4 tri, those also never acquire H2A UB?
That's right.
I mean, H2B UB? Yeah, okay.
Yeah, H2B UB, yeah.
Cool. Nice.
All right, well, my lab
one in
gets
the
to,
back.
Sorry. Thanks, Francisco.
Yeah, so those two spots we've confirmed that they correspond to the two heavily transcribed regions of the genome known as the histone locus. And so there's in Drosophila, the histones are all contiguous on one little tract of chromatin, and when they begin to get transcribed, if you stain RNA polymerase, for instance, it looks like headlights in every nucleus, and they co-localize perfectly with immunostaining of SET1. But we do see more foci later on, and we're not sure what those are.
Great. Well, I know my lab, we're going to meet with Shelby later, so we'll leave you guys away from all of the esoteric questions, and we'll just thank Shelby one more time for giving us the talk. Thanks. No raspberry though, right? Because now you have to meet with Francisco.
All right, where is he?